Hello, everyone, and thanks for joining the session today. Let's go back. My name is Benjamin Gaub. I'm Director of BD Operations, and I'm here to give you an update on uniQure. This disclaimer. So, we've been in the space of AAV gene therapy for the last 25 years, and we're proud of this legacy and continue to innovate every day. Naturally, we've had a lot of firsts in our history. We launched the first ever approved AAV gene therapy in the world, Libera. This was in 2012 and paved the way for many gene therapy products to follow. Manufacturing has always been near to our hearts, and in 2020 we opened the first commercially licensed AAV gene therapy facility in Lexington. Later that year we were able to launch a second AAV gene therapy, this time for patients with hemophilia B. And I believe this makes us the only company to have 2 AAV gene therapies on the market to date. More recently, we've been engaging with the FDA, and we got the first RMAT designation for a gene therapy for Huntington's disease. And we're now aligning with the FDA on an accelerated approval pathway. Let me give you a quick overview of our technology platform. So we have a diverse set of cargo and capsids to address various indications most optimally. On the left-hand side, you're seeing the cargo. So we have uniQure to overexpress genes. We have microRNA technology, which is microRNA-mediated gene silencing to knock down genes. And we have GoCure, which is basically the combination of those two. It's a knockdown and replace approach. When it comes to capsid, shown on the right-hand side, we're using a mix of naturally occurring serotypes for our clinical stage programs, namely AAV5, AAV9, and AAV-RH10. But we also have a capsid engineering effort where we're looking to create novel capsids with improved properties to overcome some of the biodistribution and tropism challenges of capsids today. So for administration, we're employing both systemic delivery and focal CNS delivery. And I'm just going to highlight quickly that if you have an indication where you need a lot of transgene exposure in a very particular area in the brain, then the focal CNS delivery works well as it overcomes the biodistribution challenge. It also lowers the cost of goods dramatically, and there's less systemic liability of these AAV vectors when you directly inject them. So AAV manufacturing, we believe, has been a long-held differentiator for our company. We developed an insect cell-based platform that generates rapid research material. It's scalable, and we get high production yields. However, as the environment has changed over the last few years, we decided to divest our commercial manufacturing facility in Lexington to our partner Genesen, and we retain our manufacturing facility in Amsterdam for research-grade material. Now, I want to stress that Genesen is a very close partner to ours. They actually have the same process, the same operators, the same machines as we used to use, and they continue to manufacture our commercial hemogenics batches and some of the late-stage Huntington's batches for us. So this is uniQure's publicly disclosed pipeline. We have a mix of liver-directed and CNS-directed diseases that we're going after. The liver-directed diseases are encompassed by hemogenics. This was licensed by CSL Behring in 2022. Coming behind that is a phase 1/2 asset for treatment of Fabry disease. Here we overexpress the alpha-galactosidase enzyme. For the CNS-directed programs, these are encompassed by AMT-130. This is a program where, to treat Huntington's disease. We've got an RMAT designation and are aligned with the FDA on an accelerated approval pathway. Coming behind that, we have a phase 1/2 asset to treat a familial form of ALS, namely SOD1. Here we're using a microRNA to knock down the SOD1 gene. And we have another phase 1/2 asset for the treatment of refractory temporal lobe epilepsy. And in this case, we're using a microRNA to knock down an excitatory glutamate receptor, GluK2. So as I've shown you before, it's an exciting time for uniQure. We have 4 assets in clinical development, and now I want to take some time to walk you through each one of them and give you a bit of a flavor on what we're doing here. And I'll start by Huntington's disease. Huntington's disease is a disease that is genetically inherited. It's a midlife onset, and it affects about 30,000 patients, which are diagnosed in the US. Alone, we think there's actually many more patients that will become sequenced once there's a drug available to them. The disease is caused by a CAG trinucleotide repeat expansion in the Huntington encoding gene. This leads to protein misfolding and aggregation, and ultimately neurotransmitter dysfunction. Our approach to treat this devastating disease is to use a microRNA directed against the Huntington's gene exon 1, and we're delivering this using an AAV5 directly injected in the striatum. The AAV5 we're using here, by the way, has been heavily de-risked by our hemogenics program, so it's been applied in north of 80 patients so far with no serious adverse events. So now I'm going to show you the data that got us and the Huntington's community so excited when we released it last year. I'm going to take some time to walk you through the slide. So we're using the Composite Unified Huntington's Disease Rating Scale, in short CUHDRS, to assess the patient outcome. And we found that this is the most sensitive and robust measure of disease progression in Huntington's disease patients. It's a composite score made from 4 clinical rating scales, and one of them is the total functional capacity. It basically evaluates how patients are going on with their everyday life chores, their energy levels, etc. And then there's also the total motor score, which evaluates movement of patients. And then there's 2 more cognition tests. Just to say, these are all clinical measures, no biomarkers, and so it's a quite robust measurement. For the control, we decided to use a synthetic control arm, which is made from natural history patients. And these are propensity score matched against the patients that were in our trial. So they have the same CAG repeat length, the same age, etc., to make sure these are good controls. Now let's look at the data on the top left-hand side. We have followed these patients for 2 years, and there's 9 patients in the high dose and 12 patients in the low dose. And as you can see in the teal color, we get 80% slowing of disease progression in the high dose, and we get 30% lowering of disease progression of the low dose cohort. We also find that the NFL, neurofilament light chain levels, which are a sign of neuronal death, are much reduced versus baseline at 24 months compared to natural history. We believe this is very significant data, and we're quite excited for it because it shows long-term durable efficacy with a potential for clinical benefit in these patients, something that nobody has demonstrated before in Huntington's disease. This program was started 5 years ago. We've come a long way. We have a 3rd cohort, which we're going to complete enrollment this year and give a safety update on, and we're aligning with the FDA on statistical analysis plans and CMC. We're also going to release a 3-year update on the cohort, which you see the data from in the middle of this year. So we approached the FDA and aligned with them on our accelerated approval pathway, or on the regulatory pathway. We felt they were very supportive. This is still on the old FDA under Peter Marks, but they really kind of helped us along the way and tried to support us here. The highlights from the meeting are shown here. Most importantly, we can use the existing data from our phase 1/2 studies as the primary basis for a BLA submission. For the accelerated approval, no other studies are needed. We can use the natural history data as a synthetic control arm, and we can use the composite UHDRS score as an intermediate clinical endpoint and the reductions in NFL levels as supportive evidence. So this was a big win on our end. Now I'm going to switch gears a bit, and then the remaining time that I have, I want to highlight a few programs that we have coming behind the Huntington's disease program. We have AMT-260 for the treatment of refractory temporal lobe epilepsy. This is an acquired condition, so not genetic. It affects about 1 million people in the US and EU, and most of them are refractory to existing anti-seizure medications. We found that in a lot of these patients, there's a glutamate receptor overexpressed. So there's an excitatory receptor, and the overexpression of that receptor naturally makes a hyperexcitable network. So our approach is to use a microRNA directed against the GluK2 receptor, delivered by an AV9 directly in the hippocampus to then knock down the expression of this excitatory receptor. On the left-hand side, in mouse models, you see that our treatment reduces the seizures significantly, but it also improves the general health of these animals. They're quite sick and have poor nesting and grooming behavior, and after treatment, the experiment unblinds itself. You find that the mice that are treated behave much more normal. Moreover, in epilepsy, you can actually have access to tissue from epileptic patients that have been resected. So we can slice those brain tissues and apply our treatment. So we can find that seizures are much reduced after treatment, which de-risks the path to translation. Another disease we have in our pipeline is AMT-191 for the treatment of Fabry disease. In this case, the deficiency of alpha-galactosidase leads to accumulation of GB3 and lyso-GB3 levels, and this ultimately leads to heart problems and kidney failure. Enzyme replacement therapy exists, but we feel that it has certain limitations, like poor PK and limited uptake by cells. So our approach is to deliver the human GLA protein, which will mean we have the correct post-translational modifications. And because it's expressed in this human cells and secreted by them, we think the cellular uptake is going to be better. We're applying this as a single IV injection, and it's packaged in the same AAV5 that we're using for Huntington's and also hemogenics. So what we find in mice preclinically on the left-hand side is a very flat PK profile. This is due to constant expression of the transgene, and we find cross-correction in the kidney, as shown on the right-hand side. The last one I want to highlight here is AMT-162 for the treatment of ALS SOD1. This is a very aggressive disease where patients die about 2 to 4 years after diagnosis due to motor neuron dysfunction. We're basically following the path of an approved therapeutic, tofersen, which is an antisense oligo against the SOD1, but it has a big shortcoming. It needs to be intrathecally administered once every month, which is a huge burden for patients. So we're basically vectorizing this, and our differentiation is to have a one-time treatment with a single IT administration. Good, so this is kind of over the last year and this year, the events that have happened for our company. I'm incredibly proud to be standing here today, as last year has been tremendously successful. We're able to make great improvements with the FDA on our accelerated approval pathway. We're able to demonstrate nice data on efficacy with the 80% disease progression reduction, as I showed you for the high dose. But moreover, we're able to dose the first patient for 3 additional programs, which for a small company like ours is quite phenomenal. This puts us in a good place, and we're looking to read out potential clinical proof of concept for these programs end of this year or beginning of next year. And we're going to provide an update on the cohort 3 in our Huntington's program, as well as the 3-year data, which is highly anticipated for cohorts 1 and 2. I'm going to leave you with this slide. So today in my talk, I have shown you our capabilities at uniQure, both in terms of technology and manufacturing. I've walked you through our pipeline with the emphasis on AMT-130, as this is our lead program with blockbuster potential. We're aligning on the BLA submission for AMT-130, and we're building a commercial and medical affairs organization at the moment. And I think what's also important to mention is we're in a strong financial position, so we can actually fund all of these activities and see the programs through to key inflection points. That's it from my side. Stay tuned. I hope you have a great rest of the conference.